Donc, English opening remarks will follow. Nous sommes heureux de vous accueillir aujourd'hui en présentiel et en ligne pour la 3e édition du Symposium étudiant unique. Donc, tout d'abord, quelques mots à propos d'UNIC. UNIC est l'acronyme pour Union Neurosciences Intelligence Artificielle Québec. C'est un centre de recherche à l'intersection des neurosciences et de l'intelligence artificielle financé par les FRQNT. Géré par l'Université de Montréal en partenariat avec Polytechnique, McGill, Concordia, l'Université Laval, l'Université Sherbrooke et l'École des technologies supérieures, UNIC regroupe 60 membres chercheurs à l'intérieur et à l'extérieur de la province, ainsi que plus de 850 membres étudiants. UNIC a pour mission de réunir les chercheuses et les chercheurs de premier plan en provenance de diverses institutions pour prendre part à une initiative de recherche au carrefour de l'intelligence artificielle et des neurosciences. Comprendre l'intelligence biologique et renforcer l'intelligence des machines par l'exploitation mutuelle de leurs principes communs. Être à la fine pointe de la recherche dans le domaine de la neuro-AI au Québec et en assurer un leadership international. Former la prochaine génération de scientifiques experts en matière de cerveau et de données pour répondre aux besoins croissants du Québec en personnel hautement qualifié. Et finalement, promouvoir l'équité, la diversité et l'inclusion dans la recherche et la formation en intelligence artificielle et en neurosciences. Nous, les organisateurs, souhaitons reconnaître que les terres, les eaux, où la plupart d'entre nous vivent, où nos universités sont situées et où se déroule actuellement le symposium étudiant, sont des territoires traditionnels non cédés des peuples autochtones. L'endroit communément connu sous le nom de Montréal s'appelle Tchao-Tchao-Gay, ce qui se traduit approximativement par l'endroit où les groupes se divisaient ou se séparaient dans la langue de la nation Mohawk, Gagné-Geaga. Bien avant l'arrivée des colons européens, ce territoire a été et continue d'être un lieu de rassemblement pour de nombreux peuples autochtones. Et malgré les pressions permanentes du colonialisme et de l'oppression, de nombreux Autochtones continuent d'y vivre. Avec cette reconnaissance territoriale, nous souhaitons prendre un moment pour réfléchir à notre rôle dans ces systèmes d'oppression et nous engager à apprendre la riche culture des Premières Nations de cette région afin d'être de meilleurs voisins pour les communautés autochtones à l'intérieur et à l'extérieur de Tchao-Tchao-Gay. Dans cet esprit, nous avons ajouté des liens sur notre site Internet dans la section « À propos » vers quelques ressources que vous pouvez explorer pour en savoir davantage sur les peuples autochtones de cette région. Nous soutenons profondément les valeurs d'équité, de diversité et d'inclusion. Nous sommes déterminés à faire du symposium étudiant unique un événement où chaque personne se sente incluse et confortable, et nous demanderons votre aide afin de mener à bien cette démarche. À cet effet, nous voulons souligner que le concept principal du code de conduite est le respect. Respect des origines, des croyances, de l'âge, de l'identité de genre et des préférences, du statut. Du respect à l'égard des personnes en situation de handicap physique ou mental, à l'égard des expériences personnelles, académiques ou autres, et à l'égard des différences en général. Nous souhaitons également mettre l'accent que ce symposium est un événement interdisciplinaire. Et donc, ces participants proviennent de milieux différents. Nous voulons célébrer et encourager l'aide mutuelle et la participation de tous et de toutes. Les désaccords et les discussions sont les bienvenus, mais le respect est de mise. Toute forme de discrimination et de non-respect du code de conduite ne sera pas tolérée. Maintenant, passons à l'horaire de la journée. Donc, nous avons commencé en présentiel avec un atelier. Et maintenant, nous allons poursuivre avec la session des Keynotes, qui sera entrecoupée par le dîner. Ensuite, s'en suivra la table ronde sur les dynamiques et l'optimisation en neuro-AI, et nous allons finir la journée avec les présentations étudiantes. Vous pouvez avoir accès à l'horaire détaillé sur notre site Internet. Donc, je voudrais également vous rappeler que l'événement est en format hybride et qu'il est également et que toutes les keynotes et la table ronde sera disponible sur la chaîne YouTube d'UNIC par après. Pour cet événement, nous allons utiliser l'application Slido pour les questions et nous vous encourageons tous, que vous soyez en ligne ou en présentiel, à utiliser cette application pour poser vos questions. Par souci d'accessibilité, le reste de l'événement se déroulera en anglais, mais vous pouvez toujours poser vos questions en français et nous les traduirons au besoin. Je vais maintenant laisser la place à mon collègue Zeke pour le discours d'ouverture en anglais. Merci.
Okay, so I apologize for starters to the bilingual folks here because there'll be a bit of repetition, but I would like to start things off by introducing myself. My name is Ezekiel Williams, Zeke for short, and on behalf of the UNIQ organizing committee here, I would like to welcome all of you, both in person and online, to the third edition of the UNIQ Student Symposium. So this year, the UNIQ Student Symposium is of course running in this hybrid fashion. And the theme of this symposium is dynamics of optimization and learning in brains and machines. Well, to start things off, in case for those of you who didn't get things down from the French introduction, I'll say a few words about UNIQ right now. So what this acronym actually stands for is Unifying Neuroscience and Artificial Intelligence in Quebec. And UNIQ is an organization with 5 objectives. So the first objective is to act as a kind of multi-institutional initiative to organize and collaborative research at the intersection of artificial intelligence and neuroscience, neuro-AI. The second objective is to unravel the biological kind of underpinnings of intelligence and to boost artificial intelligence through shared research into the principles of these two phenomena. The third objective of UNIQ is to spearhead research in neuro-AI within Quebec and also to act as a leader internationally. The fourth objective is to train the next generation of data and brain scientists in this province to fulfill the role that, to fulfill the need in Quebec for highly skilled personnel. And finally, the fifth objective of UNIQ is to promote equity, diversity, and inclusion in neuroscience and artificial intelligence. So before we kind of get into the more neuro-AI side of things, I would like to make a couple important announcements here. The first is that we, the organizers, wish to acknowledge that the lands and waters where many of us live here in fact, the lands and waters where many of us live and where our universities are located and where the symposium is taking place today are the traditional unceded and unsurrendered territories of Indigenous peoples. In fact, the place commonly known as Montreal is Jojage, which roughly translates to the place where the group divided or parted ways in the language of the Ganakahaga, the Mohawk Nation. Since long before the arrival of European settlers, this territory has been a gathering place for Indigenous peoples. And despite ongoing forces of oppression and colonization, many Indigenous peoples continue to live here. So with this land acknowledgement, we wish to take a moment to reflect on our role within the systems of oppression and to commit to learning about the rich Indigenous culture of this region so as to be better neighbors for the Indigenous within the communities, both inside and outside this region. That in mind, we've added a few links to our website where you can learn more about these communities and join us in this endeavor. It's under the About section on the USS page. So the second remark that I want to make to start things off is about our code of conduct. So we at UNIQ deeply believe in the value of diversity and inclusion. And in particular, we welcome and celebrate diversity of gender identity, of preference, physical appearance, nationality, place of origin, religious beliefs, cultural and ethnic backgrounds, socioeconomic status, age, personal experience in a broad sense. We are determined to do our best so that every person feels welcome, safe in this environment, and we kindly ask that you help us in this endeavor. Therefore, in order to create a safe space for everybody here at this symposium, we would like to remind you that we have all agreed upon a code of conduct, which is available on our website in English and in French. Behaviors that go against this code of conduct, such as any kind of hate speech, will of course not be tolerated, and we will take consequent actions against these. Regarding diversity, we should also mention that USS is, of course, a very interdisciplinary event, and therefore we are glad to have many participants from a diverse array of backgrounds here. That means that we are not all on the same page on all topics, and instead of being bothered by this fact, we celebrate it and we encourage mutual help and participation for effective interdisciplinary research. Disagreeing and discussing is, of course, very, very much welcome and encouraged, but discrimination for levels of education or scientific background is very much discouraged. Alrighty, so I'm going to say just a few words then about our program for the day. So again, a huge thanks to Francois Pogam for the workshop this morning on debugging in Python. As we move forward with the symposium today, we'll be having 2 keynote talks. One is about to come up, and we'll be having a panel discussion. Finally, in the afternoon, after these, we will have a poster presentation and some lightning talks available online. So I would like to remind you all, as I mentioned, that this meeting is hybrid, and therefore the talks and the panel discussion will be streamed on YouTube and will be available to rewatch again, to enjoy the fun of the symposium as many times as you like. Additionally, we have a Slido channel, and we highly encourage everyone, both in person and online, to post any questions that they may have during the day on the Slido channel. So this is how we're kind of organizing the questions at this event, just to make things run more smoothly with online and in-person audiences. So please check out the channel. I will try to write up the link to it over on the whiteboard and add your questions, and also upvote questions from other people that you're excited to have answered. Now, this kind of concludes the main opening remarks that I'd like to make. Unfortunately, I have, I think, a little bit of disappointing news to start things off. Our first speaker, Dr. Guillaume Hennequin from Cambridge, actually just had a fire alarm in his building before the start of the opening remarks. So we were hoping that maybe the 15 minutes that we spent on opening remarks would allow the fire alarm to chill out a little bit. But I guess we're just waiting on news from him. I don't know if you, hello. Can you hear me all right, Dr. Hennequin?
Yep.
Fantastic.
We're so happy
to have you. The building didn't burn down, I gather?
Not yet.
Alrighty, then I'll take a moment to introduce you, and then we'll get things underway.
Cool. Thank you.
So it is my honor today to introduce our first keynote speaker, Dr. Guillaume Hennequin. Dr. Hennequin studied electrical engineering and computer science at Supélec in France and completed a master's degree in theoretical neuroscience in Edinburgh. He went on to pursue a PhD in the lab of Wolfram Gerstner at EPFL in Switzerland, and subsequently completed his postdoc in the lab of Matti Langell in the engineering department at Cambridge. After completing his postdoc, he was awarded a faculty position at Cambridge, where he now works as a member of the Computational and Biological Learning Lab. His research group is broadly interested in the principles that govern plasticity and dynamics of cortical networks and their role in probabilistic computations, such as perception and motor control. He also maintains an active interest in machine learning, both for developing new tools for the analysis of neural data, and also for studying deep neural networks, their optimization, and what they might tell us about the brain. So I first came across Dr. Hennequin's work during my master's, and since then I continue to be in awe of the way in which he incorporates diverse mathematical disciplines into his highly impactful work at the intersection of neuroscience and AI. These disciplines include mathematical physics, control theory, probability, and especially aligned with the symposium today, optimization and dynamical systems theory. So Dr. Hennequin, we are so very excited to have you here speaking with us today. Without further ado, I'm turning the mic over to you.
Thanks for the amazing introduction, like super kind of you. Thanks for the kind words. Very sorry about this, this last minute fire alarm. Of all times and dates they could have chosen. Let me share my screen. Can you all hear me okay?
Yep. Yeah, this is perfect.
All right, and you can see my slides. Amazing. I don't think I can see any of the audience, but I'll just imagine a fantastic audience out there. All right, so actually this talk almost never happened because the email that I got as an invitation had in a subject that raised 2 red flags for my spam filter. The first was keynote lecture, which usually is enough to send anything in the trash bin. And the second thing was USS, which happens to be the name of our university pension scheme. And they've been spamming us for the past 2 years or so. Anyway, so it's nothing short of a miracle that I'm speaking here today. My lab is, as was very nicely summarized, is broadly interested in figuring out how brains compute. And in particular, we have an emphasis on probabilistic computations. So like computations that require an explicit manipulation of uncertainty, things like perception and motor control. This talk will be very much focused on perception and vision in particular. I'm going to be talking about models of V1. Now, it's clear that machine learning, as you guys probably know better than me, has made great strides in recent years, developing algorithms that perform even better than humans at object recognition, at scene understanding. There's been amazing work at the interface between neuroscience and machine learning in this space, in particular in kind of linking the type of representations that emerge in these deep networks to the activity that you might see along the ventral stream in the brain. Now, while we learn a ton about representations and algorithms through this type of work, I think the idea that all of this is somehow implemented in the dynamics of brain networks kind of tends to slip through the cracks. And this is very much the core focus of my lab and the focus of this talk today. So we're interested in the dynamical regime of those circuits. And we've been attacking this from 2 different angles. One that has to do with phenomenology and one that has to do with function. So on the one hand, we tried to build kind of minimal models of cortical dynamics that explain as much as possible of the phenomenology of brain dynamics, what we observe in the actual brain. And the other one is about kind of training RNNs or optimizing RNNs on functional objectives of the sort that I just mentioned, perception, motor control, and then trying to relate them back to what we observe in the cortex. And so today I'd like to tell you how we've been able to actually connect the dots between these 2 families of approaches. And ideally, what you'll walk away from my talk with is the idea that there is a simple canonical model of cortical dynamics that performs that's capable of performing sampling-based probabilistic inference for low-level vision on the one hand, and recapitulates a lot of the phenomenology of cortical dynamics on the other hand. So it turns out that the crucial piece in the puzzle of linking these 2 different types of approaches for us has been to consider the variability of neural responses to sensory stimuli. And so I'd like to start my introduction with a brief recap of what we know about variability in the brain and why it's so important in this endeavor that we're in. So cortical variability, the thing about the brain is that quite unlike your favorite ResNet or AlexNet or whatnot net, sensory cortices appear like hopelessly unreliable. If you show the same stimulus again and again and again across many, many trials, and you record response of, for example, that one particular neuron in cat V1, you'll find out that the response is essentially different on every trial. Now, unless you're doing dropout or something, this is not something that typically happens in your deep networks. So that inevitably makes the study of perception much more difficult than it could be otherwise. But, and this is my one attempt today at being kind of remotely inspirational, in the middle of difficulty lies opportunity. And more seriously, though, I'd like to convince you here that there's 2 opportunities that are afforded by what we observe about variability in V1. And they will essentially form the basis for my talk. So the first opportunity is that variability actually has a rich phenomenology. There's a rich patterns of variability that are observed in V1 and other sensory cortices that actually provide very important information about how we should go about modeling brain dynamics altogether. So the first aspect of variability is that, well, it exists, right? This is something you find when you record the brain. And in fact, the very existence of variability is exactly what spurred the entire field of balanced networks. So those of you who are into the business of computational neuroscience and network dynamics will know very well what I'm talking about. There's a whole family of network models for cortical dynamics that rely on excitation inhibition balance. I'll return to that in a minute. And the very initial development of these models was meant to account for variability in the most primitive form. Now, much of this variability is actually shared. That is, it's kind of variability that is coordinated across neurons, and that presumably reflects kind of a network level phenomenon. People might say it's actually low rank. It has low rank structure, and I guess you'll hear more about low rank networks with Patrick Desrosiers tomorrow, I think. More importantly for us, variability is actually modulated heavily by things like attention, brain states, and in particular, stimuli, sensory stimuli in particular. So this is kind of a figure taken from a famous 2010 paper showing that the Fano factor, which is a standard measure of spike count variability across trials, when you repeat the same stimulus again and again 100 times and measure the variability of the spike count distribution in a sliding window for a particular neuron or across the entire population, you find that systematically, and this is true for V1, MT, V2, V4, even for the motor cortex during motor planning and execution, systematically there is a drop in variability following stimulus onset. So there's something so peculiar about this that it seems like it's something that really any model of cortical dynamics that takes itself seriously should actually reproduce somehow. Not only is variability modulated by stimuli, but it's modulated in very fancy ways, in ways that actually depend on kind of the tuning of mean responses of these neurons to stimuli. And I'll return to that later on in the talk. And altogether, all these phenomena really ought to constrain our mechanistic models of brain dynamics. Okay, so we'd be really fools not to consider them in building theories. Now, the second opportunity that variability of responses brings with it is the fact that perhaps it's actually used for something. It might actually be a key component of computation in the brain, and if it is, then again, this is going to provide very important cues about how dynamics serve computations in these particular brain areas. So there's been an endless debate about just from a pure information processing point of view, whether noise variability is hurting you, and if it does hurt you, whether it hurts you just a little bit or whether it's terrible. But this has been studied to death by a lot of people, and there's a couple of bold people that went as far as proposing that actually variability might be an essential substrate for probabilistic computations, and in particular in the representation of uncertainty in the brain. And this is very much kind of the working hypothesis that we will take in this talk. And I refer you here to a couple of papers by Matthieu Langel, who's my postdoc advisor then and a colleague now, who has worked a lot on pioneering these ideas. I'll tell you more in kind of about this hypothesis in more detail later on. Okay, so this is essentially how I'm going to articulate my talk. I'll start by showing you how the variability of responses in the cortex can be used to constrain theories of cortical dynamics. I'll show you how we built a minimal model of cortical dynamics that accounts for a lot of these phenomena that I've just explained. That's going to be roughly the first part. The second part is going to start from the premise that indeed variability as observed in the cortex exhibits signatures of probabilistic computations. And this, again, is kind of bootstrapping on work by Matthieu Langel and others. And we will address the link between these two things, and we'll be training recurrent neural network models that belong to this minimal model family that I will be talking about in the first part. We'll be training these networks on probabilistic computational objectives. And then I hope to convince you that once you do that, actually there's a host of dynamical phenomena that are also observed in the cortex that just emerge as a result of training these models on these tasks. All right, so onwards. I'll begin now with a very brief recap of EI balance, because this is so central to the first part that I have to take a slight detour and kind of tell you about the origin of this and what's really at the core of these models, for those who perhaps are not familiar with this type of modeling. So as I said, stick an array of electrodes in the cortex, record from many neurons simultaneously. You'll observe that each of them fires irregularly with roughly Poisson statistics, and fairly asynchronously as well at the level of the whole population. So as I said earlier, this observation is really what kind of spurred an entire line of investigation into network models. What sort of network model can account for this type of variability? The first that was, well, perhaps not the first, but one of the first that was proposed, and that certainly was by far the most influential afterwards, was the one proposed by Carl von Weswig. And this is also an opportunity for me to kind of pay tribute to his scientific work. Unfortunately, Carl passed away about 2 months ago, I think. And he's really kind of has left us with this beautiful theory of EI balance, which I think beautifully exemplifies kind of the very first part of the quote that I had left out earlier by Einstein, which is out of clutter, find simplicity. I think this model that he developed really, really embodies this principle. So what he proposed together with Chaim Sumpolinsky was a model of a randomly connected network made of split populations of excitatory and inhibitory neurons. And he showed that using self-consistency arguments that the model can operate in a regime where if you take a particular neuron within the network, that neuron is going to receive a net excitatory input that is very large, and indeed many, many, many times the spiking threshold, which would be the upper limit of that gray band here, just above rest. Okay, so the net excitatory input is huge, of order root k, where k is the number of synapses onto each neuron, or at least the order of magnitude of that number. Likewise, there's going to be a net inhibitory input that is also a very, very strong negative of order minus root k. Now, the network can kind of conspire to make these two sources of input dynamically cancel each other and track each other in such a way that you're left with a small remainder, which is 0 on average, but kind of inherits the order 1 fluctuations of the E and I input separately. So you get order 1, as scaling with k, right? You get kind of k independent fluctuations in the net input, which then drives sporadic and occasional action potential generation in that cell. Okay, and that kind of explains the sort of irregular and asynchronous firing that we observe in the cortex. And again, as I said, this theory has been extremely influential. It's probably formed the basis of many, many tens of later developments in terms of models. Now, there's a but. There's a big but, especially for the purposes of this talk. These are the buts. So the first two, I'm not going to linger too much on them, but the third one is very important. The big problem with tight balance, as I've just described it, is that it leads to linear input-output behavior. So unless you resort to arguments based on nonlinear dendritic integration, then you're going to run into this problem, which is that if you double the input, which is the firing rate of that external population drive here, if you double that, then all that happens is that you double the output. Okay, and so that's completely linearized, despite the fact that every neuron is itself very nonlinear. As a whole, the population conspires to respond linearly to its stimulus. So that's a problem because if we could train deep linear networks to perform object recognition, then I guess we'd know it by now. But there's another important problem that's related to variability, which is that these balanced networks exhibit kind of incompressible or kind of irreducible Poisson variability. Okay, so there's no way that changing the firing rate of the external drive here is going to modulate the Fano factors in these networks. And these Fano factors are almost universally just about 1. Okay, so that conflicts with the observations that I made earlier. This conflicts with the fact that the cortex tends to operate with super Poisson variability in the spontaneous regime and with under Poisson variability in the stimulus evoked regime. Okay, so that's that's kind of a big blow in the theory of balanced network, in my opinion. So taking these, especially the first problem on board, Ken Miller and Yashar Ahmadian, who were mentioned on my first slide, well, actually, they were just there by picture, but I will mention them later. They came up with, together with Dan Rubin, they came up with this kind of revised theory of balanced networks, which they call the stabilized supralinear network, SSN. I'm going to spend a bit of time describing that model because it's the very model that we're going to use to explain all the rest in this talk. Okay, we're going to make it stochastic later, but the basis is the same. So what is this model? It's essentially the same model as the classical, balanced network model. We've got two interacting populations of excitatory and inhibitory neurons. They're connected with recurrent weights, such that the dynamics of the voltage, which is represented here as a vector across the population, is driven by a couple of terms. The first is a return towards the resting potential on some timescale tau. And then there is two additional sources of drive. The first is external input, H, and the second one is recurrent input. And this is given by a linear mixing of the presynaptic firing rate. Now, the relationship between, so you should think about this voltage, by the way, as kind of the spikeless voltage. So think about the voltage as the voltage you would normally measure with the spikes, but with the spikes removed and maybe low pass filtered on a time scale of 30 or 40 milliseconds. Okay, so we're talking about kind of coarse grained membrane potential dynamics here. Now, the relationship between the momentary firing rate in those 30 millisecond bins and the average membrane potential in those same bins is something that has been measured in CAT-V1 by David Förster's lab. And it has the form of a threshold power law nonlinearity. So for very low membrane potential, there is no response. And for very high, and as the membrane potential increases, then the firing rate grows quadratically, cubically, quartically. It's some exponent that is somewhere between 2 and 5 in CAT-V1, anesthetized, I think. Now, this, again, that looks very much the same as a standard balance model, but the main thing that Yashar and Ken did was to revise the assumption that those net excitatory and inhibitory input would be very large. Okay, so they work in the regime, they analyze the regime where these inputs would not be of order root K, but of order 1, or maybe a couple of times the spiking threshold, but not much more than that. And they showed in these beautiful papers that when you do that, then the network acquires nonlinear response properties. And I'm just going to give you an example in this particular two population model. As you increase the input drive, you can see that the network transitions into, from a regime of superlinear integration of these inputs. And in particular, in the firing rate, you've got something that looks like a quadratic response integration early on. And it transitions into a regime of very strongly sublinear integration of inputs for larger inputs. Okay, so there is something really fancy happening at the network level. Again, each neuron in its own would just respond like the gray lines. Okay, this is a bunch of disconnected network when there's no interactions. And the network interactions in this with this particular threshold nonlinearity, threshold quadratic nonlinearity, kind of conspired to bring about this kind of saturation for larger stimuli. This is just one example, but in that Rubin paper, Ken and Dan Rubin explored the consequences of this dynamical regime for V1 dynamics, and in particular, they looked at steady state responses, and they found that they could explain a whole bunch of response nonlinearities that V1 is known to exhibit, in particular, things related to normalization and like things related broadly to responses to multiple stimuli. What happens when you superimpose multiple stimuli in your receptive field? And they also made a bunch of novel predictions that were kind of, that you wouldn't have guessed a priori, I can tell you that, that they were actually able to verify experimentally in ferret V1. So altogether, I think this model is a very compelling model of V1. It, as Ken likes to say, it explains a lot with little. There's really not much that goes into the model. You put in kind of realistic assumptions about the form of the input-output relationship in single neurons. you put in the fact that neurons come as excitatory and inhibitory neurons. And as long as you set up the network in such a way that it can dynamically stabilize, then you get all these nonlinearities that emerge. So that's all really cool. And what we did next was we wondered if the same model could explain aspects of response variability like I've introduced earlier. So to explore that, we just took that same model and we made it stochastic. So we just add an external source of noise, which is going to drive a cross-trial variability in single cell responses to stimuli. So in this simple two population model, this is just a mean fit model with just two populations, E and I, we found that indeed the stimulus here, H, tends to quench membrane potential variability, at least past some thresholds. So what happens here, and I can give you a very intuitive, I'm not gonna elaborate on the math because I don't have time for that, but I'm gonna give you kind of the main intuition for what's going on here. So here's the input that goes in steps. And here is the membrane potential, stochastic membrane potential response of these E and I populations. What you can see here is that for small h, what happens is that the membrane potentials are small because there's nothing that drives them, which means that the neurons are sitting in the very flat part of their input-output relationships, which means that effectively, if you look at the Jacobian of the dynamics, if you linearize the dynamics around the set point, you find that the effective couplings are going to be W times the slope of this input-output relationship, which is nearly zero, which means the couplings are very weak, which essentially means that each unit processes input noise privately. And therefore, the noise that you see here at the bottom just simply reflects the noise that you put in. Okay, so this is just a baseline amount of variability that you put in. Now for medium H, what happens is the network is driven upwards. The network is driven towards steeper parts of its nonlinearity, of the single cell nonlinearity, which means we have stronger couplings. And the network can enter a regime where excitatory and inhibitory feedback kind of have comparable size, in such a way that actually the noise is allowed to reverberate through the dynamics. Okay, and that leads to this kind of inflated amount of variability in the middle. As the stimulus grows further, the couplings become yet stronger. But what happens now is that, and this is kind of almost universally true in these networks, if you want dynamical stabilization, you have to set up the network in such a way that inhibitory firing rates grow faster than excitatory firing rates. Okay, and if you are in this regime, then you'll find that soon inhibitory feedback dominates over excitatory feedback, and that the net effect of that is that it quenches fluctuations. I guess tomorrow you'll have, or maybe today, I don't know, you'll have a tutorial on the mathematical analysis of dynamical systems, and you might hear about eigenvalues and that sort of stuff. And so this is very much the way that we analyzed these effects in the paper. So check out our supplementary material if you want to know the details. But that's kind of the main intuition behind this kind of nonlinear effect of variability quenching. So we think that actually the cortex might be normally operating in a spontaneous regime somewhere near the middle here, okay, where you've got firing rates on the order of a few hertz, and you've got enough excitatory feedback that drives reverberation of noise, and therefore you would get kind of super Poisson variability in these networks. And then any stimulus that comes from this baseline can only quench variability as is observed in the cortex. All right, I hope you guys are still following. I can't see any of you. I don't have any feedback. I'm just gonna soldier on and assume that everyone is fine with what I'm talking about. All right, so like any self-respecting theorists, we had to build a ring model of V1 at some point, and this is what we did next. So we wanted to understand if this model could also capture more the fine-grained structure of variability across trials that's observed in V1. So for that, we kind of build a canonical kind of ring model of V1, which is meant to represent really a hypercolumn of V1, where you have essentially all preferred orientation in one given population. So conceptually, we lay down the neurons on a ring. And again, it's a balanced network, so we have split E and I populations, each of them, again, kind of are arranged on the ring. The position on the ring is going to end up representing the preferred orientation, the preferred stimulus orientation of that cell. As you guys may know or may not know, I don't know, but cells in V1 are very much tuned to a particular orientation in the stimulus in the receptive field. The connectivity is going to take the form of standard bell-shaped connectivity profiles that fall off with the difference in those preferred orientations, as schematized here. And the stimulus is, again, also going to be a bump of input centered here around 0 degrees, but obviously the whole thing is rotational invariant. So just to fix things, we just put the stimulus at 0 degrees in those slides. The stimulus is going to be dependent on contrast. So we model contrast as a simple scaling of this input bump. And at 0 contrast, there is, again, a baseline, there's essentially this baseline input that instates a spontaneous activity state in the network. Okay, so this is what activity looks like in this model as a function of the preferred orientation along the ring. So these are just the cells, and this is time. And you see that before stimulus onset, activity kind of wanders around. There's kind of large fluctuations in the firing rates. And then when the stimulus comes up, like it very much pins this kind of bump of activity at 0 degrees where the stimulus lies, okay? And we can analyze variability in this model. We actually computed phenofactors, assuming that on top of these kind of firing rate dynamics that we model, we would also kind of emit spikes stochastically as a doubly stochastic Poisson process. This is a fairly standard assumption to model spike counts. And we find that phenofactors are indeed quenched, very much like in the experimental data at stimulus onset. So we go from super Poisson variability to Poisson variability. Here we can actually go below Poisson in this model. And the interesting thing that we noticed was that this drop in variability is actually tuned to the stimulus. So it happens in a more pronounced way to those neurons that respond best to that particular stimulus. Okay, so the orange neuron here compared to the brown neuron that drops less. Okay, so so this is also true in membrane potentials, by the way. And again, this has been measured in V1. There's beautiful measurements of membrane potential fluctuations in V1 and how they are quenched by stimulus onset. And again, this is done in a particular, there's a fine-grained structure of this variability quenching that really depends on the stimulus tuning. So we wondered if we could actually go back and reanalyze data from monkey V1, and we took a data set that's publicly available by Alex Ecker and in Andreas Tolias's lab. And we performed this analysis where we looked at how phenofactor depended on the preferred orientation of the cell. So this is the spontaneous regime, where obviously it's not supposed to depend on the preferred orientation of the cell because there is no stimulus at this stage. And when the stimulus comes up, we found like a very pronounced U-shaped modulation of phenofactors in V1. And this is very much in line with what the model predicts. The model predicts a U-shaped drop in phenofactor, and as well as a U-shaped drop in membrane potential standard deviations. But we don't have data. There's not enough data on that to actually draw these curves. Okay, so this is all great. This is, well, I think it's great. I don't know if you think it's great now, but this is kind of a model that's both simple and accounts for these fancy phenomena. But I think what's even better is that we could use variability data to rule out another candidate model that had been proposed previously to account also for these kind of patterns of variability in the sensory cortex. And this model is based on attractor dynamics. You guys may be familiar with attractor dynamics. This is also a very standard canonical model of brain circuit dynamics. The idea is you have embedded in the connectivity and the dynamics, you have multiple attractors, multiple kind of regions of state space towards which the dynamics tend to be attracted. And the idea is that before stimulus onset, there's nothing that biases you particularly to any of these attractors. And so across trials, you might be found in one or the other attractor before the stimulus comes up. And so this is what these trajectories represent. So we've got a greenish trajectory and an orange-ish trajectory that unfold in time. And you can see that before stimulus onset in this multi-attractor model, the activity tends to kind of switch between attractors and therefore cover quite a big part of the state space. Okay, and that leads to a lot of variability before stimulus onset. And of course, when the stimulus comes up, then it kind of pins you to one of these attractors, and that reduces variability. So that's a perfectly sound model of variability reduction. That is kind of mechanistically very different from the way that the stochastic SSN operates, which it operates in a regime where at any given time there's only a single stable attractor point. Whether you're in a spontaneous regime or in a stimulus-driven regime, there's only ever a single point of attraction. Okay, so qualitatively, it's a very different regime. So we figured that actually we could rule the multi-attractor dynamics out. And that was by looking at the fine structure of pairwise correlation. Like so far, everything that I've told you about was about single cell responses. Okay, so we looked at how single cells behave in their mean responses and in their fluctuations. But now we're going to look at how pairs of cells co-vary as a function of the stimulus. So this, let me just walk you through this particular graph. So we partition cells in the SSN model. We partition pairs of cells according to whether they have a similar preferred orientation, whether they are close to each other on the ring, or whether they are on opposite sides of the ring. Okay, and we made this kind of 45-degree bands partitioning of these pairs of cells. And then we look at how, in the evoked regime, at high contrast, how the pairwise correlation in their noise responses, so response minus the mean, right? So the pairwise correlation in these noisy fluctuations, how does that depend on the orientation of stimulus relative to the average preferred orientation of those two cells? Okay, so that's a bit of a mouthful, but essentially you can think about this as kind of a way of taking slices through the full covariance matrix that dictates the all pairwise correlations in a network. Okay, so these are two particular slices that are that behave qualitatively differently. You can see there's a U-shaped modulation along the diagonal. So for similarly tuned cells, again, those cells that are similarly tuned and happen to be best responding to that particular stimulus orientation are most suppressed in their correlations, and those that are similarly tuned but not tuned to the stimulus are less suppressed. And what we find interestingly is that for cells that are orthogonally tuned, then there is almost no modulation with stimulus of their pairwise correlations. And again, we can, you can look at our supplementary material. We go into quite some depth, kind of explaining why this happens mathematically. Now, in the attractor model, it turns out that the dominant mode of activity or mode of fluctuations in an attractor model, in this case, it's a continuous ring attractor model of the type that was proposed here, and that was originally proposed back in 1995. This ring attractor model is dominated by kind of sideways motion of an activity bump that's otherwise rock stable. Okay, so you've got, the stimulus drives a bump of activity whose height and width is rock stable, and the main source of a cross-trial variability in this model is the sideways motion of the bump around the stimulus orientation. And that in particular gives rise to these very pronounced negative correlations between cells that are oppositely tuned. And so we went back to reanalyze the monkey data, and we found that the data really supports the SSN and rules out the attractor model. So all in all, I guess, so this concludes the first part of my talk. And I would say that, we have again, a simple model that explains quite a lot with very few assumptions. We have the basic assumptions that there is a threshold Paolo input-output relationship between voltage and firing rates in each neuron. Then we have this assumption that we have split populations of excitatory and inhibitory neurons. And we have the further assumption that these populations balance each other in such a way as to maintain dynamical stability in the network. Apart from that, all of these phenomena kind of arise fairly robustly. All right, so now we're going to move on, and I'm not going to explain how variability exhibits signatures of probabilistic computations, but I'm going to tell you exactly in what way we believe that variability is used for probabilistic computations. And then I'll walk you very quickly through the training of these SSN models that we've done on probabilistic objectives and how we can recapitulate aspects of V1 dynamics that we haven't accounted for yet. All right, so what's the main idea here? So the main idea is, comes from behavioral observations, mostly in the sensory motor domain in particular, that brains construct internal models of the world. In particular, they construct internal models of how the environment behaves and how the environment impinges on our senses. Okay, so for those of you who are in the machine learning world, like think density modeling of sensory inputs. This is something that our brains are thought to be doing, and in fact are known to be doing, except we don't know how. So the main idea then to investigate this line of thoughts is to build a generative model, or to kind of take a good generative model for the type of processes that we're interested in. In this case, it will be low-level vision or local vision. Again, we are interested in the behavior of a hypercolumn in V1. So a column that's really responsible for processing visual input in a small corner of the visual field. And for low-level vision, then a good candidate generative model, something that explains the structure of natural image patches very well, is the so-called Gaussian scale mixture model. So without going into too much detail, that model says that those small patches of natural images arise as linear combinations of basis functions, which we call projective fields, each of them being weighted by kind of the intensity with which that feature is present in the image patch. And these AIs are called latent variables. And the whole thing is further scaled by a kind of shared contrast variable Z, which is drawn from some prior. The model assumes a Gaussian prior, a multivariate Gaussian prior over the activations, and some gamma prior over Z. But these details are really kind of not consequential. Okay, so then the brain's job is to perform, well, learning of this model, obviously, but that's outside the scope of this talk. But more importantly here, it's to perform inference. So it's to invert the model in a Bayesian way and to condition on the retinal input that we're currently receiving, and to infer a whole posterior distribution over these latent variables, or these intensities A's. Okay, and this is just a slice of what the posterior might look like. Actually, it's a representative slice, because for when Z is sufficiently determined by the stimulus, then the posterior over A happens to be a Gaussian posterior. And so we might have, covariations under the posterior between latent 1 and latent 2, and they each have their own means. And so this is just like an illustration of what one slice through this high-dimensional posterior might look like. The hypothesis that we're going to work with here, which was originally proposed by Mate Langel and colleagues, and even, in fact, earlier by Apo Ivarinen, I think. The hypothesis is that V1 represents the posterior bisampling. Okay, and what I mean by this is that there's going to be one cell in V1 that's responsible for coding for, whose activity represents our belief about, our momentary belief about the intensity of latent 1. There's a second cell whose activity represents latent 2, and then the network kind of conspires to generate activity trajectories that map out the state space in such a way that it spans, it covers each portion of space in just the right proportion dictated by the posterior density. Okay, so we could just superimpose these two cartoons and have network trajectories kind of literally overlap with that Gaussian posterior that I'm showing on the left. Okay, so that's the main assumption, and you can see how this assumption, just this assumption, actually allows you to bridge kind of probabilistic computations with what we observe in the brain. On the other hand, like the noise variability we observe in the brain in a completely normative way. And in fact, this is what a lot of papers by Mate and colleagues have done in the past. Okay, but in this talk, we are primarily interested in the dynamics that might give rise to this particular type of inference, of sampling-based inference. So we're interested in training. So what we're going to do next, and this is really going to be the work of Rodrigo Echeveste, who was a postdoc between me and Mate a couple of years ago, And he trained models within the SSN model class that we've been discussing so far. And he trained them to perform amortized inference under this generative model. And what I mean by this is that even though we've known for a while, and this is also in some of my past work, that if we just have a single stimulus and a single posterior density, P of A given S, then we know how to set up kind of realistic looking network dynamics that will sample in just the right way, right? And in fact, we also know how to do it in a way that they sample very fast, which is important in this context. I'll return to that a bit later if I have time. The difficulty here is that we want the same network sharing the same set of parameters to be representing the posterior accurately for any stimulus. Okay, so we want to really perform amortized inference in such a way that you feed the network with any pattern of retinal activation. It goes into the network through what we call feed-forward receptive fields, and then the recurrent dynamics of that balanced network up there has to be just right in such a way that the variability exhibited, well, the mean, first of all, but also the variability of those responses is going to reflect sampling from the right posterior distribution. Okay, how am I doing with time, by the way? I lost track of when I started because of this fire alarm. Like, do I still have 10, 15 minutes?
Yep, I think we still have about not quite 15.
Okay, 10.
We have 5 minutes.
5 minutes. Okay, cool. So I'm going to skip some of the details of how exactly we train this model. This is just to show that training is successful. This is a bit the kind of, this is what took us most of the time, but probably what you care the least about to make sure that we could train these models in such a way that it works. We trained these models on only 5 different stimuli, in this case, at different contrasts. And we find that indeed the posterior, which is shown here for different values of the contrast in terms of mean responses and response standard deviations, are very nicely matched by the network responses that we trained. This is just to show, again, we capture the dependence on contrast of the posterior variance or standard deviation here, as well as the dependence on the posterior mean, of the posterior mean. It's not specific to training examples. This was all for training stimuli, except in between those dots here, which is interpolation. But it's also, we can also extrapolate, and we get a very good calibration of the network on test stimuli that have kind of more than one orientation content in them. So this is really just to say that training is successful, and now we can actually go ahead and kind of look at how this network behaves. So the first thing that we asked is that under a sampling representation, time is actually of the essence, right? So we want, because the only way to get an accurate picture of your posterior is to just wait for samples to be produced, wait for these trajectories to cover the whole space in a way that's compatible with the posterior density. But for this, you just need to wait. There's no other way, right? Unless you can parallelize multiple chains. But if we do it the way we do it, then time is really at a premium. And we found that the trained SSN actually solves this by very much narrowing the autocorograms of its responses. And that's so, so this is just the autocorrelation of these activity fluctuations at different contrast levels, shown by different shades of red here. You can see hints of oscillations. I'll return to that in 30 seconds. And kind of the point I want to make here is that it's hugely more efficient than an off-the-shelf sampler, which you might have taken from the machine learning literature called Langevin sampling, which is a type of sampler that relies on so-called reversible dynamics. This might speak to some of you. And because it relies on these kind of random walk that are fully reversible in time, it just has to slow down massively. And you can see that the autocorogram of this Langevin sampler is terrible. Whereas the network here is able to speed up its dynamics by a mechanism that I'm going to show you next, in such a way that it can even process the input noise faster than disconnected cells would normally. Okay, so just the connectivity of the network that's been trained kind of instates a dynamical regime of very fast fluctuations. That's the bottom line. So it does so actually by breaking reversibility, but I'm going to skip over that in the interest of time. You can just read all about this in the paper. It turns out that in a nutshell, the network is implementing a type of algorithm that's actually known as non-equilibrium sampling, something that violates something that's known as detail balance, which has nothing to do with EI balance, by the way. This is something that actually machine learning practitioners have been coming up with to speed up their MCMC sampling algorithms. And so it's kind of very interesting that the trained SSNs actually comes up with these type of strategies. Okay, so now I'm going to just spend 2 minutes going through the types of dynamical phenomena that actually emerge in these trained networks. So this is just a very quick recap of what I said earlier. This is an SSN, so we expect these kind of tuning curves to emerge, where this is the mean firing rates as a function of the difference between the preferred orientation of the cell and the stimulus orientation. We get this kind of tuning curves, but more importantly, we get factor modulation, very much like what we have in the data. So again, this is data from Monkey V1. This is the optimized network. And as a control, and this is going to become important in 30 seconds, as a control, we also trained a network to reproduce posterior means. So we trained them on reproducing the mean responses dictated by the posterior, but ignoring the variances. So we did not train this network to actually represent uncertainty at all. Okay, all it had to do was to represent the maximum a posteriori, or the posterior mean actually, which is the same in this case. So this is going to be an important control, because everything that I'm going to show you next, it would be great if this really was specific to the training objective that we used, and in fact it is. So here's the two main phenomena that emerge in these trained networks. The first is that we have gamma oscillations in the optimized networks. You can see these power spectra have a nice peak in the gamma band, and more importantly, so this is observed in V1. This is very well known. But what's really interesting is that the peak, the peak frequency of these gamma oscillations is actually stimulus dependent, contrast dependent in particular, and so it actually grows with contrast. And this is exactly what people have found in Monkey V1. Importantly, it's not there in the control network. The control network that is not optimized for uncertainty representation just doesn't produce any gamma oscillation whatsoever. Now, we also observe like funky transient responses, so what we call on-responses. So this is again the firing rate time course around stimulus onset, which is this black bar here. And you can see that the response tends to overshoot massively and then kind of go back to what it has to be for the posterior mean. Okay. And this is again something, sorry, this is the network, and this is also something you observe ubiquitously in V1. And this is again something that is actually not showing up in the control network. So again, these arise specifically from the sampling objective, not from the constraints. So it's not seen at initialization. When we initialize the SSN with random parameters, it really emerges through optimization on that sampling objective. It's not seen in networks like the one on the right, trained on mean responses only. It's also not seen in a network trained on mean responses and trained to specifically keep the variance fixed. And it's not seen in networks trained on means plus variances, but not on covariances. So it's really, really specific to the full sampling objective that we've been using here. And finally, and I'll finish with this part with this, so Rodrigo did a lot of analyses, which you can find in the paper, actually not in the supplementary, but in the main paper, to kind of really get to the bottom of, why are these dynamical features actually showing up in these trained networks? Like, why are these dynamical features essential to sampling? And the bottom line is that, number one, oscillations are here to improve the mixing time. The mixing time is again, jargon from MCMC, the MCMC literature, but it's basically the time it takes for the posterior to be well represented by those samples. Okay, so how long should you observe the trajectory before you get a representative picture of your posterior? Obviously, if you just do random diffusion, then you're gonna need to wait a long time before you get good coverage. If you just trace an ellipse, which is not anything but like a random walk, then you'll have a very good picture of your variability in very little time. Okay, and these networks essentially perform something a bit in between. They are still diffusing, but they are diffusing in a way that oscillates, and that helps with mixing time. Now, transients overshoots, they improve what we call the burn-in time, which is the time that it takes for samples to become representative of the posterior distribution in the first place. And again, you can find all of the details in the paper. It turns out that overshoots in the model are tuned as they are in the data. Okay, so I'll try to wrap up now. So what I've shown you today is two things. The first is that we can use variability phenomena as a way of constraining our models of cortical dynamics. And I've proposed a very simple model, the SSN, or the Stochastic Stabilized Supralinear Network Model, that captures a lot of this phenomenology. And then we've kind of connected with dots with probabilistic computations by explicitly training SSNs on probabilistic objectives and closing the loop with experimental data by showing that you've got dynamical phenomena like oscillations, transient overshoots, things about detailed balance as well, like EI balance that I haven't been able to show here, but that are also captured. And a bunch more predictions that are actually up for grabs if you're an experimentalist, which I don't think there are many in this room, but just in case. And so with this, I'd like to close and thank again a lot the organizers for having me today. And I also look forward to the rest of the workshop, which I hope to be able to attend today and tomorrow.
So I'm not sure how well you can hear it there, but there's lots of applause. And I apologize about our sensory deprivation set up here for the speaker on Zoom. Definitely one of the downsides of this format. Would you have time, Dr. Hennequin, for a couple of questions? I know we're a little bit over time.
Sure. Yeah. Fire away.
Awesome. Let's see. So the top question throws us back a little bit, or the top voted question throws us back a bit to earlier in the talk. And it asks, I'm assuming the network topology used, this is with regards to EI balance network. I'm assuming the network topology used was an Erdos-Renyi random network. How does different network topology affect the results of EI kind of stability?
All right, good question. So I'm guessing the question refers specifically to this part originally. So this is this whole theory that Carl developed was indeed an Erdos-Renyi kind of randomly sparsely connected network. This is nice because you can do a lot of mathematical analysis. But then since then, I mean, a lot of people have actually, one of the drawbacks that I brought up here is the fact that in these Erdos-Renyi networks, actually they end up, the whole population ends up behaving as one unit, or maybe two units if you just consider E and I neurons separately. But the entire population of excited neurons just behave as one functional unit. The entire population of inhibited neurons behaves as one functional unit. And you're using millions of neurons to just basically compute a two-dimensional, to effectively reduce that to two-dimensional dynamics. And so you've got, macroscopic chaos at the level of single cells, which means that single cells, again, on their own, they don't do anything interesting. It's the whole population that that performs the computations. So a lot of people have tried to fix that precisely by imposing different types of network architectures. One of them is actually the ring model that I mentioned later. So like people have built stacking network models with ring topologies. I did a bit of that as a PhD student, and you can find that maybe in some corner of my PhD thesis if you look carefully. But there's been a lot by Brent Doran and Robert Rosenbaum and others who've specifically kind of analyzed the conditions for the stability of these networks. So what happens, for example, if, here you've got distance-dependent connectivity on this ring. What happens, under what conditions are these networks stable as a function of the width of the excitatory to excitatory connectivity, E to I to I to E, and that sort of stuff. So there's a fairly comprehensive literature, which I couldn't summarize in just a couple of sentences, but do get in touch with me by email if you want more pointers, I guess.
Wonderful. Thanks a lot for the explanation. So seeing as we are over time, I'll just ask one more question, and I'm going to target this one to the second part of the talk where you discussed sampling. So I'm curious, I know a lot of this work regarding sampling is, of course, kind of spreading out your inference for a stationary stimulus over a little time period. How can you speculate about how things might change if you didn't have a single fixed stimulus, but you had a temporally varying stimulus?
Yep. So in this case, the computation that you need to implement is called filtering. And this is something that some people that I know are working on. So you might want to check out the work of Jean-Pascal Pfister, who also used to be a postdoc with Nate, by the way, way before me. So, I mean, I don't know if the question is about more specifically about speculating how the SSN might be able to capture this. So this is actually a tough one because we haven't actually, I would need to think for more than 5 minutes. But what I can point you towards is the fact that there is some literature on kind of the neural implementation of Kalman filters. There is a beautiful paper by Sophie Deneuve and I think Alex Pouget, or was it Peter Latham? I can't remember. Maybe someone at Gatsby back in, I think, the early 2000s, I think, showing how, I don't know if I'm using too much technical jargon here, but like the problem you just described is known as a filtering problem in probability, in kind of probabilistic machine learning. We've got observations that come as a stream, and you know that your latent variable is not fixed, but keeps moving in time. And in this case, you need to keep track of the posterior in a dynamical fashion. And so in linear Gaussian networks, this is known as Kalman filtering. In linear non-Gaussian, non-linear non-Gaussian networks, such as the generative models such as the GSM, you would need to do more fancy type of inference. And the bottom line is that we haven't tried, but this is a very interesting project, I would say. Kind of see how the SSN could be trained on that sort of dynamical objectives.
Very cool. Well, I think we're going to have to wrap things up, as much as there are many more questions I would like to ask about this exciting talk. So with that, thank you so very much for joining us, Dr. Hennequin. It was a pleasure.
Thanks for the opportunity, and have a good conference.